like this is like the nesting dolls of introductions. We're just going to keep introducing people till we're finished in an hour here. Well, thank you all for coming. It's my pleasure to introduce the speaker for today, Dr. Dave Dallas. He is also fairly new to the nutrition faculty. It's coming up on a year now, right? Yep. And kind of a homecoming of sorts, as he's a native Oregonian, was raised in Pendleton. He did his training at Rice University in Houston as an undergraduate in public health, and then went on to UC Davis, where he entered the nutritional biology program and started some of his work there on human milk proteins, and then transitioned into a postdoctoral position there in food science and technology. And so I think that combination of perspectives and his background really made him a really interesting fit for the nutrition program here as we think about kind of the next steps in our vision for the future of nutrition within the context of public health. And I know he's got a lot of exciting ideas about how to, advance the field in general, which is a story for another day. But today he'll be talking to you about his research in human milk proteins.
Thank you, Emily. That's like the nicest introduction. Okay, so can you guys hear me? The mic is plugged in. Yeah. Someone is, they're filming it, so I have to officially louder for you. Okay, so yeah, I'm really happy to present today. It's been about a year since I started, and so it's kind of giving you an update about what we've been working on in my lab. And some of my lab members are here, so Cern and Vero and Yuki and Melinda. I have a really great team of people. And the other thing I was thinking about for this talk today was just that I guess my goal for this is to tell you about it, but also to try to convince other people to do more research in the field of breast milk and milk nourishment, basically. So whether that is from a public health perspective or other kinds of perspectives, I wanted to see if we could think about different ways to do more in this area. So, but anyway, so my main goal for my research, is the mic sounding good, or is it, everything's good? Okay, so my main goal for my research is to improve the nourishment of premature infants. And I'm looking at it from the perspective of protein, and because protein is so important for their health. And the ways that I'm doing this right now are using mass spectrometry to look at what protein fragments are being created. Vero has been doing a lot of work looking at what enzymes are actually cleaved, what enzymes are actually present in the milk, and how they are affecting the infant's own digestion. And then we're doing some bioinformatics to try to predict which of these pieces of proteins are functional in the baby and could have some biological relevance. So that's kind of what we're doing right now. And my overall goal is to try to move that now into how that affects the gut health and the health in general of the infant, and try to look at more overall picture of protein digestion. So if you've seen my talk before, this will be somewhat of a review, but it's very important to me. So how did I become interested in milk? I became interested because when I was a grad student and I was studying nutrition, I began to see how complex things were. I began to see that there are thousands of foods in our diet, and there are thousands of molecules within each one of them. And it becomes really hard to know what each of those things does for your health. And so, intimidated by that, I was like, oh, milk is actually really a nice, easy place to start for me, because milk represents the tug of war between the mom and the infant. It represents over 200 million years of evolution to feed the baby. So then we can assume that everything in it is beneficial for the infant. So if I look at proteins, or I look at oligosaccharides or fats, I know that no matter what I look at, it's going to be beneficial for the infant, because the mother is breaking down her own tissues to produce each piece of the milk. And so if it's not beneficial, it doesn't get passed on through the gene pool. So So that's why I think milk is really cool, and that's why I study it, and that's why I think it would be cool for other people to study it as well. Yeah. So yeah, milk has evolved from just like actually it was a sweat gland secretion, they think, and each piece has been added over time to make this complete nourishment system for the neonate. Milk proteins are really important because they provide the building blocks for growth and immune function and all the things that proteins are important for. And so I wanted to study that component, but when I looked into the literature, I found that milk proteins are actually much more complicated than you would expect. So if milk proteins serve only as a source of amino acids for the baby, then it would make sense that milk would produce a large amount of one or two different kinds of proteins that provide a nice balance of amino acids for the baby, right? But milk doesn't do that. Milk produces thousands of different proteins, and every year there's papers coming up with increased numbers of proteins that are being produced there. And those proteins come from all across the genome. So milk was developed relatively late in evolution compared to all the other different organs and systems in the body. So the genes for milk have come from all across the genome. So they're basically borrowed from different systems. So the question that I always had was, why would milk do that if it could just create this, balanced source with a couple different proteins? Well, it's because these proteins are functional beyond their source of amino acids. And so more and more research is showing that different milk proteins have specific functions. Like lactoferrin is a protein that is very strongly antimicrobial in milk. There's immune modulators. There's things that help absorb calcium, like caseins. But of course, milk is not kept intact. And so most of the research on milk proteins and their functions, trying to figure out what all these proteins are doing, they look at the intact protein. They try to see, what does that thing do as it is? But really, that's not what happens in the baby for the most part. For the most part, you have digestion happening. So my question was, what happens to those proteins when they go into the baby? And what do those fragments, those different pieces of protein do inside the infant? Like, are they still functional there? That's kind of the question that I'm looking at. And if you go into the literature, there's a lot of really good information about this, sort of. So it's kind of weird information because basically dairy companies, people who are making cheese and yogurt and all these things have for a long time thought, oh, I have this product. Can I make some sort of functional claim about it? So they've taken the proteins in there and they've put them through a lot of their favorite assays. And those things are things like, can we lower blood pressure of old people, for example? Do we have blood pressure reducing peptides? Do we have things that are antimicrobial? What can we say about our product, basically? But because of that, we now have hundreds, thousands of different peptides, perhaps, that have known functions. So this is kind of a good place to start for us, and we can see sort of the range of possible functions that could be produced from milk. And now I'm taking it to a different perspective and looking what is actually produced in the baby. So in thinking about that, I realized that premature infants are really important for this question because premature infants, it looks like they don't digest protein as well. So if they're born early, they may not have had enough time to completely develop their intestine and their gut functions in general. So I wanted to see, is protein digestion different in these babies? And does that difference in digestion release different peptides with different functions in those babies? And that could lead to different health outcomes. So then in order to look at this, what I did was I partnered with the neonatal infant care unit in Sacramento at UC Davis. And currently I'm expanding here in Oregon to work with Randall's Children's Hospital and OHSU at their NICU. But basically what we do is collect samples to monitor digestion in the baby. So we get breast milk samples, we get gastric samples if they're tube fed, and we pull a sample back up from their stomach after a certain amount of time of digestion. And we can also collect stool, urine, and we're looking into potentially blood sampling. But that's kind of how we do that. And so the next step was how to look at what's being created in the gut. And so I had to sort of create this new area. It's kind of a niche of proteomics called peptidomics. And basically, we had to figure out a way to extract all of these peptides from milk, all these pieces, and then to be able to identify them. So I don't want to go into all the details about it. You know, there's an extraction technique. You're removing the fats, you're removing intact proteins, removing sugars and salts and all that stuff, and you get just this pure peptide mixture. And then we do mass spectrometry on it. And we've used a bunch of different mass spec techniques over the years. Now we're using an Orbitrap, which allows for, some of my slides will say like that we've identified hundreds of peptides, and now we can identify thousands of peptides. So basically, the faster the instrument is, the more precise it is, the more you can identify. And so it uses really highly accurate mass. It also breaks the peptides into pieces so that you can sort of solve a puzzle of what the peptide sequence is, basically. So there's thousands of different sequences, and in order to solve that question, we use different software to be able to analyze what peptides are present. So the question was originally, does milk contain peptides? And we didn't really think that there was going to be peptides in milk because we thought, okay, this is just the starting place. Milk proteins are intact until they enter the baby where they start to get cleaved. But what we found is that there are a ton of different peptides already being produced in the mammary gland. And originally we had like 300 peptides. Now we are showing thousands of different peptides being released from milk proteins. And they also are pretty much the same between mothers, you know, between a range of mothers and across time. There's a very high consistency of what things are being produced. So then we thought, okay, what's responsible for these things being released inside the mammary gland? And if you look in literature, people in the dairy industry have looked at this for a while. There's a bunch of different enzymes in milk, and they looked at it from a perspective of fouling their products. So if you have a cow with mastitis, for example, it might produce a lot more protease enzymes, which can lead to degradation of the proteins in the milk, which makes it not very good for making cheese, basically. So if you have degraded casein, it won't form cheese very well. So that's kind of been known about. And so what we wanted to do was investigate what happens with human milk, basically. And there's this whole array of different enzymes here. There's proteases, there's the precursors to them, zymogens, there's activators, inhibitors, all these things that form this complex web that seems to be controlling the level of peptides and the kinds of peptides that are released. So our first way of looking at this was to say, okay, can we use bioinformatics? Can we take the sequences, the specific sequences, and where they are cleaved, and compare those with the known cleavage patterns of enzymes to predict which enzymes had created them, basically. So we're trying to reverse engineer what had happened, basically, in the milk. And so we did that, and we found that some of these enzymes that were known to be in milk are functional and are cleaving these proteins within the mammary gland. And so then what we've been doing now, and what Vera has been working on, is taking this from just a prediction, which is nice, and it's an interesting way to use peptidomics analysis, but moving it to actually measuring those enzymes specifically, how active they are and what's their abundance, basically. So she uses a substrate assay, basically. So you have a specific sequence of amino acids that matches really closely with the cleavage pattern of a protease. So each protease has a different specificity. It cleaves at different sites. It likes different places. And so we pick different substrates that match those. And then when they cleave, they release a fluorophore. So you can see how much of that enzyme is active in the solution. So we've got this mixture. We've got probably 8 different enzymes that we're measuring, and then we're doing a lot of ELISAs to see what's there, basically. So what we found was that a lot of the enzymes that we predicted to be in the milk are actually active in the milk. So they're not just there, but they're working. So milk is actively digesting itself within the mammary gland. By activity, we can rank them in terms of how active they are. There's a few of these for milk scientists that would be surprising that are more active than we thought they would be normally. So we're showing for the first time the activities of a lot of these enzymes, basically. We also found that there are a lot of inhibitors enzymes, and they're present in high levels. So you've got this mixture of things happening here. So previously, going back a little bit, we wanted to see what kind of proteins are being cleaved in the milk. And we found that it's specific. So certain proteins are releasing lots of peptides, whereas other ones are releasing very few or none. So like certain proteins like immunoglobulins and lactoferrins are not really releasing peptides, but caseins and other proteins are releasing a lot of peptides. So there's some specificity into what's happening in the mammary gland. And this is kind of an ongoing thing that we've been working on, is what functions do the peptides have? So, so basically at Davis, I had been working on this for a while, gathering the results from literature that I was talking about with people looking at cheese proteins and all those things to see what functions they have. And basically, we've taken that now to create a very exhaustive database. So that's what CERN and Yuki and Rob have been working on. Basically, they've kind of a heroic amount of effort, honestly, but they mined 2,800 articles from literature, found like 254 that have active peptides, and they created this like very carefully curated database that shows what do we know about milk peptides, like what is like the whole body of literature about them, so that from now on, when milk researchers look at this, they can just use the database instead of having to like go to all these hundreds of different articles, basically. So we're created a resource for the community. And then we can use this to analyze what peptides we find in the milk and in the infant's stomach and the other parts of the gut against this list to help us find out what things might be functional. And so there were some other databases that were non-specific to milk, or this one is just antimicrobial. And basically, we made sure that it also includes everything from these databases as well. So it's very exhaustive. And the functions are things like antihypertensive, antimicrobial, anti-inflammatory, immunomodulatory in general, opioid activation. So there's a lot of things. And we looked at milk from all the different species that were available, so from human and goat and bovine and all these different things. And so now we're able to like map what these peptides are, how active they are, and how they are in literature. So we can map, like this is for antihypertensive peptides. We can see from what part of the protein are most of these coming from and how active they are. So as you go down, they become more and more active in terms of their effect. So we're using that to sort of summarize what's happening in milk. And like for antimicrobial activity, we're looking at how frequent these peptides that are antimicrobial are across the sequence of the protein, and how it compares between like a bovine milk protein sequence and human milk protein sequence. So this can help guide people to identify new peptides, find areas that are worth more exploration, that sort of thing. And we have taken this list and we search our own peptide lists for how homologous they are, so how close the peptides that we find are to the sequences in our database. And we find, hundreds of different peptides that are present in the milk or in the stomach that are matching with functional peptides, basically. And then another thing we did was, like, if the peptide was present in bovine milk, but it wasn't found in another kind of milk, like human milk, we're using our homology tool to map those things onto each other so that we can identify many more peptides in different species that were not identified before. So there's like 1,000 sort of new peptides that were not previously identified in those species. So now you could say, okay, this bovine milk cheese has all these ACE inhibitory peptides. Now you could say that goat milk cheese has it as well. So it's kind of a little bit of diversion, but yeah. So now what we're trying to do is predict what peptides are functional. So not just what peptides match with known ones in terms of their sequence, but look at the structures of those peptides to try to figure out whether or not they're likely to have a function. So CERN is looking at a bunch of different programs and looking into building his own programs to make these predictions. So some of these predictions, we're using them to look at like across a protein sequence. You can see like where the peptides were present. This is kind of being weird. Is that normal? So you can see where the peptides were across the sequence, so we can map that. And then we're mapping it to sort of red would be like the areas of more increased frequency of predicted to be antimicrobial. So we combine of all these different programs to find out which ones are the highest likelihood so that we can then do the next step, which is testing them. So we're trying to identify the new antimicrobial peptides that are present there. And so basically what we're working on right now is picking those, like looking at different programs to pick the best ones, synthesizing them, and then we'll test them against an array of bacteria that we've picked out because they're relevant to mastitis, so inflammation of the mom's mammary gland, or to the infant's gut health, or sort of things related to the infant's throat, basically. So like where it would interact with the peptides in milk, basically. So we've looked at how peptides differ between term and preterm milks, and we found that in general, you have higher enzyme levels in preterm infants, in the milk of mothers of preterm infants than you would have in the milk of mothers of term infants. So they release more peptides. And they release, we showed they release more from specific proteins like casein. By prediction, it looks like their enzyme activity is higher in the preterm. And it's interesting, they release peptides from the same places, just in different frequencies. So even in the preterm, still seems to be a controlled pattern of what enzymes are doing to those proteins. So some stuff that we've been doing lately, Vero is looking at what enzymes are present and active across a group of preterm infants that we had collected, ranging from really early preterm infants, so like 24 to 26 weeks gestational age, so sort of on the border of viability, 24, to later gestational age infants, so 27 to 32 weeks, and then looking at how the milk changes from a sort of earlier time point, 2 to 26 days, to a later time point, 30 to 47. And these time points are a little bit wonky, but it's been really hard to collect the samples in the perfect way. which can happen with human studies. And that's partially why Melinda is working on redesigning a study to make more tighter controls and all that sort of thing. But in general, most of the milk enzymes stayed pretty constant across time and across gestational age for the milk of preterm infants' mothers, except for a few of them. So plasmin activity looks like it decreases a little bit across postmenstrual age, and kallikrein activity or kallikrein concentration can increase a little bit in late days of life for just the early gestational age infants. So there's a few interesting things there. I think mostly what I take away is that there's not that much change between these infants, but they all have active enzymes and lots of milk protease activity. Okay, so now getting to digestion, we want to know how much is digested, what peptides are released, and what enzymes are doing it, basically. So if you look at a peptidomics perspective, we actually did not think that there would be much digestion in the stomach of these babies. And that's because they produce very little acid, and the main protease responsible for digestion in our stomachs is pepsin. And pepsin requires acid to become activated and for it to stay activated. So if you have a baby who doesn't produce very much acid, you get like a pretty neutral gastric pH where the enzyme couldn't be active. But you can see here in blue is the gastric peptides, and you can see a ton of new peptides are released compared to the intact milk. And this is in term infants. You can see that the number of peptides increases significantly in the milk or in the stomach, so there's clearly digestion happening there. And you can see that it affects specific proteins, so things that there's no red over here, so things that were not digested in the milk now are released in the stomach, so like lactoferrin, some immunoglobulins, alpha-lactalbumin. So now we're taking it here, we've been looking at what enzymes are doing this. So is pepsin working? Is it the milk enzymes? What's happening? And so first of all, we see that the pH does decrease in the premature infant stomach, but it's still pretty high, like 4.6, is very suboptimal for pepsin's activity. So we would expect very little pepsin activity, but we see a lot of protein digestion happening. So we first just looked at, is digestion happening by some different methods. So we looked at lactoferrin by ELISA. We can see it reducing from milk to the gastric. And that is commensurate, or at the same time, we can see increases in the peptides from that protein. So it's, again, showing digestion. Same with immunoglobulin by ELISA. The intact protein decreases in the stomach, and the peptides released from it increase. So we're kind of like two different ways to show that digestion is happening and looking at how much digestion is happening. And then some of those antiproteases that we found in the milk are also being digested, and we can see that with our ELISA tests. We are seeing decreases in antitrypsin and antiplasmin and antithrombin and plasmin serine protease inhibitor, and you can see increases in peptides from those enzymes as well as matching. When you get to the stomach, the activity of the proteases does change a lot. So you can see increases in elastase activity, increases in cathepsin activity, a decrease in kallikrein. So clearly there's a lot of change in what's happening in the stomach. And it's not just about the pH increasing. So you're going from a pH of 8 in milk, or 7, somewhere between 6 and 8 in milk, it ranges, going to the stomach where it's like 5. So how would that affect the enzymes? Well, in this case, you're going away from this pH optima of this enzyme, going from what it would be like more neutral to more acidic, but the activity still increases. So we don't know exactly what that's caused by, but it could be from the reduction of the antiproteases because they start to get digested. So we're kind of looking into explanations of why these things are happening. We found that milk doesn't contain any pepsin, which is what we expected, but there is some amount of pepsin secreted in the premature infant's stomach. And the changes in activity in the stomach are dependent in some cases on the specific group of infants. So the later gestational age infants, like they're premature, but they're a little bit later, at later days of life, increased levels of elastase activity. So there's some variation in enzyme activity with time, basically. Oh, but I think the most interesting thing to me is that if we compare the amount of activity that we calculated for pepsin versus the amount coming from milk proteases, the vast majority of the enzyme activity was coming from the milk proteases. So they are it's showing that they are controlling digestion inside the infant's stomach. So yeah, so now we still need to compare term versus preterm digestion. It can be challenging to get term infants that are in the NICU that have the tube feeding, but we're working on that. And we're moving into looking at what happens in the stool and the urine, what peptides survive there. And then they would have more potential to interact with, the lower digestive system and different receptors. So the new study that we're doing, mostly with Randall's Children's Hospital, we're going to look more carefully at what happens to infants across gestational age. So do the earliest premature infants have the most trouble with digesting? And tracking them across time in the hospital so that we can compare one infant at one time point to 7 days later, to 14 days later, to 21 days later. So we can have a more continuous time course and a really well-controlled study. So that's what Melinda is working on. And we're collaborating with a couple, a doctor, a neonatologist there, and the lead dietitian there, Andy Markle, who actually went to OSU. So yeah, again, we're doing that. And then we have some project going on with a gaggle of interns right now. So we're looking at donor milk, and if these proteases are important for infant digestion, then how does the way that we handle them for donor milk affect their activity? So nowadays, donor milk is becoming more and more common. Mothers will send in their milk banks where they will mix it, they'll pasteurize it, make it so that it's safe for the babies, and then they ship it to hospitals for premature infants to use for their nourishment. So we want to see how the different ways that they could treat the milk to eliminate the bacteria affect those proteases. So this group of interns is working on that, and we're also looking at what happens if you freeze your milk, and how long you freeze it. Does that change the proteins, the peptides, the enzymes in the milk? And ideally, we want to try to find if we can figure out better ways to handle donor milk to increase the quality of it, and et cetera. And then just some new directions, things that I'm thinking about, so I've been wanting to do it for a long time, and I think maybe I can do it now. But basically, it is to be able to trace overall protein digestion, to measure that in the infant. And what it would be is feeding moms with a stable isotope in their food so that the proteins in their own milk become labeled, intrinsically labeled with a stable isotope. Then feeding that milk to the baby along with a free labeled amino acid. So you have one attached to the protein and one that's free. And then measuring a small amount of their blood after 2 or 3 hours of digestion to see the ratio of the free amino acid and the protein-bound amino acid that makes it into their blood as a way to measure how digestible the protein is. And that way we can measure what are the overall differences between premature infants and term infants, and between early gestational age and late gestational age, and between formula versus breast milk or donor milk versus breast milk. So that's kind of something that I really want to get into now. And I'm trying to find people who know about these kind of things at OSU to help me with that. But yeah, so that's what I have for today. I thank very much my really great team of people and interns and collaborators at Davis as well. So thank you. I'm looking forward to your questions and comments and ideas for collaborations, everything. So thank you. I blew you all away, huh? No, just kidding. Probably talked too fast.
I have a question.
Yeah.
So I think in one of your slides you talked about this milk being from healthy mothers. Do you know if any of these things change if you have a mother with obesity or diabetes, if the nutrient composition is changed a little bit, if it's drastically different?
So I think gestational diabetes can affect the amount of like lactose and other sugars in the milk. It could probably, but I don't know about how it affects protein. So I guess the answer is I don't know yet, but it could be interesting to look at. Yeah.
Related to that, do you have any sense of what the role of the cause of preterm birth is in the various outcomes that you're observing? So if there's, I don't know, there's bunches of different things. I wonder if those causes themselves might function to change the outcomes that you observe in infants. Or with regard to the quality of the breast milk or other characteristics.
Yeah, I think that those are definitely things that would be really interesting to look at. But we haven't yet. That could be a good future question to look at for sure. And I was thinking, I really liked this concept of doing the stable isotopes and stuff to see what sort of the number of digestibility is, because I think that could be easier to translate to some of these other kinds of questions where it's you have the effect, a specific effect, and it's easier to relate to the specific change in digestion. Whereas I think I've struggled with peptidomics because there's so many different values that you're measuring, and so it makes it a little bit challenging overall picture.
Well, I understand extremely little about what you do, but I think that it might be very easy to extract from medical records, from people that you already have, some other variables that you could, put into some models or at least compare across.
Yeah, we were thinking about how much antibiotics was the baby fed, different variables about the mom, and so for the future study that Melinda's working on, she's collecting a bunch of different data like that. But I haven't thought about the cause of the preterm birth yet, so maybe we need to do that. And obesity and diabetes.
Mine is sort of a follow-up to that, because I was interested in that too, what else might be going on, especially with the mother and then maybe with the baby as well. Very premature. And I wondered, how stable are your findings, so that how large of a sample size do you need to use for some of those in order to feel like you really found your findings?
Yeah, so with the enzymes, it seems like we need a sample size of 10 per group is what we've been looking at to see changes if there are changes. So it's okay, it's doable for the populations we can get. With peptidomics, we found it harder to see changes, and I think it's because there's so much variation in what could happen. And that's why I'm also drawn to the stable isotope method, because it would be a single measurement that would be pretty precise, that maybe is able to help us see those differences better. Yeah.
Can you say a little more about stable isotopes?
I can try.
Just say the approach, what you have in mind.
Yeah.
Because maybe I can help with somebody. Because I wasn't sure from what I heard, you want to have the mother endogenous synthesize its own protein.
Yeah.
And then go from there.
Yeah.
Or you want to have something that's already synthesized, a stable isotope of a protein, and digest it, and then you go from there. There are various methods where you want to use radiated method all the way, just
exactly.
more that you have in mind.
So what I found in papers, no one has really done this per se, but I found people, getting milk proteins in cows to be highly, intrinsically labeled. There are some studies from the 90s where they did it with humans, and they fed them labeled phenylalanine, so the phenylalanine was labeled in the proteins. And because you synthesize milk really rapidly, then it gets incorporated there more than in the other tissues. So it's easy to label. And then, so then putting it in the infant and seeing how much is absorbed. So does that, that's what I'm thinking about, but I need to know more about like which ones to choose and the timing and everything. So I need to do some like pilot testing. But yeah, there was another paper where they used a spirulina protein, so it's this algae basically. And they did it with some kids that had cystic fibrosis. And they were continuously fed, and they were able to measure this ratio, which is doable, but it would be much nicer to have it with the actual human milk protein because it's obviously differently digestible than some, standard protein. Yeah, definitely. It would be awesome to chat. Yeah.
Hey, David. In follow-up to what Tammy was asking about, the kinetics are not a simple system. It's my understanding that there's some points of control as far as what proteins are expressed, never mind what's on board. Correct? It's not just like if I put a certain percentage of phenylalanine in mom's diet, that percent is necessarily going to simply be expressed or be in play as far as what is made for breast milk.
Okay, so you'll have, like, different moms will probably have different levels of intrinsic labeling, right? Maybe it will vary between people. And also different proteins will have different amounts of label. Is that what you're kind of saying?
Yeah, I'm just thinking it seems like it'd be a little bit more complex. But through different fluid distribution.
Yeah, so I think the key thing for this is just giving enough so that you have a good level of enrichment. And then once it's in the infant, finding out what time to collect the milk or to collect the blood from them. So trying to figure out when has it been fully digested and reached the bloodstream. But then using, if you measure it before and after and like getting the proper ratio of that compared to the free amino acid that's produced, I think that you have all the information that we will need.
And my second question is, have you contacted the industry players as far as Ross Labs? Are they interested in this kind of work? Are they doing this kind of work? Do they have?
As far as I know, no one is doing that.
Okay.
Because
it seems a way that they would be interested in trying to, their motivation would be, how do I make
more
formula closer to the adaptive post-expression physiology that you're talking about is some of the magic of mother's milk.
Yeah. I think that would be a really interesting thing to start looking at. Yeah. Mm-hmm. Yeah.
I had another question about the timing when you were looking at the digestion. So you said that your divisions were kind of odd, but to me, when you're looking at a 47-day-old baby, so anything within that range of what you're measuring, and then say you look out to 6 months or even beyond, do you know how that changes over that longer trajectory? Because there's a lot of thinking now around how long to recommend, mothers breastfeed their babies, and if there are benefits beyond a certain point, or if cow's milk is really a reasonable substitute once their own digestion matures to a certain point. So I don't know if you have any thoughts on kind of beyond that early.
Yeah. Well, I guess I was kind of limited before when I was looking at the gastric sample, because obviously that's really hard to get if you're an older baby, because it's usually just, the first month or two post-birth. But using a method with stable isotope, I think that could be used to look at later on, and then could start looking at those questions. But I know that there's ramping up of the different digestive enzymes in the infant's digestive system, there's papers that seem to show, or not, there's papers, our papers seem to show that the enzyme digestion kind of decreases a little bit over time, at least in terms of peptidomics, but we haven't gone out to 6 months. But I think what you'll see is the decreasing contribution from the mother's own enzymes and increase in the infants. So it would be interesting to follow it further out like that.
It'd be interesting to know too the comparison with cow's milk, because right now that's a recommendation to give cow's milk at 1 year, that you can give cow's milk at 1 year, but I don't think there's a lot of really good research into why and how and what impact that has on developed child's milk.
Yeah. The other thing is a lot of formulas are now shifting to giving free amino acids or like hydrolyzed proteins. It's like they call it like something like easy on the tummy or something. So it's like very digestible proteins. But then there's this question of, could something be too digestible? Because if you have just free amino acids all the time, you're not stimulating your pancreas to produce proteases. So there's some studies in rats that it shows that if you do that for a long time, you end up getting some atrophy of your pancreas. So you may be setting them up for more trouble digesting later in life by doing so. So maybe it's not as simple as just like, let's make it all super digestible. But all right. Any other questions? Okay. Well, thanks so much, everybody. It's been really fun. Thanks for all